All right. So first of all, let me thank Nita and the rest of you guys for inviting me to be the first speaker of your Aegis of Science Journal Club. I think it's a good initiative. I hope it grows and expands, and eventually you'll get probably more. I want to say qualified persons, with bigger reputations on.
But you are a whole PhD. You have a whole PhD and a
Yeah,
whole
I
doctorate.
know.
So you are the youth, some kinda.
Yeah,
So,
well, it's a little caterpillar in the game. Eventually, you'll get big people out there. Anyway,
yes, yeah.
and then today is Women in Science Day, all right? And I think now obviously there's more women in science than men, right? But that's a good step. That's a step in the right direction. But anyway, let me begin. So I want to talk about my research, and my research is broadly based on microbial bioprospecting, right? And that aspect is a pretty specialized aspect in microbiology, and it fuses so traditional microbiology with some biotechnology, molecular biology, and a lot of chemistry, right? Essentially, what I do is discover and develop novel antimicrobial compounds, or compounds with some sort of some sort of antibiosis activity, right? And so this what you're seeing on the screen here is basically what the mantra is in bioprospecting, all right? And that chemicals essentially determine how nature interacts with each other, right? Because nature itself is a series of chemicals, and their interaction would determine an outcome, right? So that's like what we do. We look at chemical interactions in nature. So one molecule influencing another molecule, and we adapt that to solve human issues, right? So, and mainly human medical issues. All right. So a pretty easy example that I use for biochemical prospecting for when I give in this talk is essentially the plant on the screen is, I guess, well, for a more interactive crowd, the plant on the screen is thyme, right? And thyme produces a compound called thymol, right? And thymol, we've adopted thymol to use in certain cosmetic and what's that? And I guess pharmaceutical uses too, right? And thymol is the active ingredient in Vicks, the rub, and Listerine, the mouthwash, right? And that is essentially what we do. We take molecules produced in nature and give them a function for human use, right? And how that works is that, like I was saying, if you examine or if you observe nature closely, you will see that organisms use chemicals for their own advantages, right? So the example, the picture I have there is insects predating on a plant, but the plants themselves, when being fed upon, emits volatile compounds into the air. And those volatile compounds could range in function. So those volatile compounds could be detected by plants nearby, and the plants nearby then starts to produce their own chemical defenses. So they start to produce their own compounds that if the insect now migrates and starts to feed on plant number 2, they could die, right? Because the plant now produces a toxin or insecticidal compound, or something that would make the leaves taste bad, right? And most of us would know that when we cut grass, you get a freshly cut grass smell, right? And that smell that we get is volatile compounds that the plant produces to warn its neighbors then that there is a threat. So it senses the weed whacker as a predator then, right? So hopefully that concept is easy enough to understand, right? And what we would do now is that when we observe insects feeding on a plant and then the insects dying, you would assume that that plant produces some sort of an anti-insecticidal compound, right? Somebody just drew up on my screen.
Yeah, who is drawing?
That's fine, that's fine. Right, yeah. So so like I was saying, so we adopt natural molecules for human and I guess plant use and whatnot, right? So some sort of beneficial use. And for microbial bioprospecting, especially in Trinidad and environments like Trinidad, is pretty good because what occurs is that our geographical location is pretty good for microbial growth and development. And then we get a lot of different microbial species or high diversity in our environments. And then in Trinidad as well, we have a lot of different niche areas that would allow for speciation to occur. And because our climate is pretty stable and with high humidity and rainfall and whatnot, we get a lot of microbial growth, a lot of microbial evolution as well. And microbes have the ability to share genes, right? So they could give and take genes from each other that would allow for adaptations and whatnot. So the relationship is the higher the biodiversity in a particular area, you have a higher biochemical diversity as well, or chemical diversity. So the more living organisms you have in an area, the more chemicals you have in that area as well, because each organism produces or has its own chemical fingerprint, right? And that's why it's particularly important to preserve your organisms, right? Your biodiversity, right? Because not only is biodiversity a good thing for natural environments, it's also a good thing for humans because you have a higher chemical fingerprint to possibly exploit, right? So my specialty is on microbes, and microbes essentially dominate Earth, right? They're about 4 billion, so they're the oldest life forms on Earth. They're about 4 billion years old, and they exist everywhere. So even in environments where you didn't think microorganisms could survive, they do, right? And not only they do survive in those environments, but they thrive there, right? They account for about 60% of Earth's biomass. 3% of your own human or the body's biomass is bacteria. And they have you have more microbial cells than you have human cells. So the thinking is that you are more microbe than human, right? So my research is shifting towards this non-traditional form of compound isolation or compound identification or bioprospecting, right? Traditionally, in labs, so microbes could 1% of microbes could be cultured in labs, right? So that we call a traditional culturing of microorganisms. So when you explore environment and you collect samples and bring them back to a laboratory environment, and you try to isolate these microorganisms, less than 1% of them would grow in the conditions that we have in labs, right? So that means there's greater than 99% of microbes in the environment that have not been discovered. We know they're there because of genetics, but we cannot physically grow them. Therefore, we can't study them and manipulate them or whatnot, right? But over the past 10 to 15, probably 20 years now, there's a shift towards independent or culture-independent methods for compound isolation or bioprospecting, right? And that's metagenomics. And metagenomics is particularly interesting in that we bypass that limitation of laboratory culture, right? And we target the genetics or the genomics of the microorganisms living in an environment directly, right? The genes or the genomes of these organisms would code for the compounds of interest, right? So the compounds that give these organisms some sort of advantage in their environment is all encoded for in their genes or in their genetics, right? So by bypassing that culture step and going directly for the DNA in these areas or in these microbes, we could bypass that, and that's known as metagenomics. Right? The avenue we're looking at now is a relatively new form of gene sequencing called long-read sequencing, right? So there are two main platforms, the PacBio and the nanopore systems, which are able to sequence long reads of DNA, right? And without getting into too much detail, we usually combine both the long read gene sequencing with the short read gene sequencing, or the traditional gene sequencing known as Illumina platforms, and then we overlay those things. The end result is to get the sequences of these genes, right, that would code for the compounds of interest, right? And the genes that are responsible for compound production is known as biosynthetic gene clusters. So in microbes, compounds of interest or compounds that confer some sort of bioactivity, right, are found in these clusters. And gene clusters are essentially amalgam of genes found in a particular area of the genome, right? And these genes, they serve different purposes. You have your core synthetic genes, or the genes that are responsible for the parts of the molecules, but then you have regulatory genes and transport genes and whatnot. But the thing is, in microorganisms, they're all found in one particular region of the genome, right? And you have key characteristics that you could look for in these sequences that you would be able to identify where a gene cluster begins and where it ends, right? And it's pretty well worked out now, where you could predict the compound that is produced by the genes that are encoded in the gene cluster, right? So we have enough genetic information now that, like I said, you could predict what compound is going to be produced by that particular gene cluster. So then you could predict if the compound is novel. And then if you know the structure of the compound, if you predict the structure of the compound, then you could predict bioactivities to that compound, right? So we know, for example, antibiotic compounds have certain characteristics or certain chemotypes. If the compound that's being produced by a biosynthetic gene cluster resembles one of those, and you could sort of predict the bioactivity, right? Anyway, so I know, sorry, I know I was talking there, but anyone has any questions so far? I'm gonna switch directions now. I'm just giving you a little bit of a overview into what is my interests currently.
I have a question, actually. In terms of gene sequencing, I saw you showed a couple methods that are currently used. What do you guys use at UE? And also, is it done in-house, or do you do gene sequencing abroad? How does it work?
Yeah, that's a good question. So we outsource the gene sequencing because to run and operate a gene sequencer is a little bit expensive. The reagents and whatnot, and the reagents have a particular lifespan or shelf life. And I don't think there's enough sequencing occurring at UE in that high demand to warrant in-house sequencing at the moment. Now that might change because those nanopore machines and whatnot are becoming a little bit more robust, benched up machines. And so eventually, hopefully, we could get some funds that would allow us to purchase our own sequencer, which would then probably accelerate our discovery, right? But that requires some sort of a push, I guess, and some sort of financial backing. But right, sorry, just to answer your question, it's more outsourced. It's definitely outsourced at the moment. So we have a couple of companies that do it for us relatively cheaply. So my research interest lies in antimicrobial resistance, right? So antibiotics were discovered early in the 20th century, and it saved millions of lives due to infectious diseases, right? So infectious diseases are those caused by microorganisms, right? And because we live in a microbial dominated world, it's quite easy to get an infection from a microorganism, right? Be it fungal, bacterial, even viral, right? But I'm going to speak about bacterial infections here specifically. So antimicrobial resistance, and I guess this crowd is pretty scientific, is where a bacteria becomes resistant to an antibiotic that that was designed to treat that infection, right? And resistance occurred pretty quickly after antibiotic use on the market. So penicillin came onto market, I think, in the late 30s, and resistance to penicillin was observed maybe a year or two afterwards, right? The thing is, resistance isn't something that we invented. It's not a human problem. It was always there because the microbes that produce antibiotics have to be resistant to that antibiotic, right? So microbes produce antibiotics to kill off their competition, but the producing cell has to have some sort of intrinsic resistance, right? Or else it'll kill itself. The thing is, because microorganisms could share genetic material quite easily, bacteria that was susceptible, so that the antibiotic would have killed, would have picked up genes that would have made it resistant. And then when humans use antibiotics indiscriminately, then we kill off the populations that would be susceptible, and we allow those small populations that was resistant to grow and flourish. And now we have more resistant bacteria in circulation or in population than we have susceptible, right? So almost all the bacteria that we have now in hospital settings and now in community settings are resistant to one or more antibiotics. And that's just a fact, right? The graph I have up on the screen is a prediction graph. It shows the number of deaths that would occur directly from antimicrobial resistance by the year 2050, unless some sort of intervention occurs, right? And you see the two largest circles, so the two greatest proportions is in Asia and Africa, right? Antimicrobial resistance, apart from mortality, also results in billions of dollars in losses in productivity, right? So when you get sick from an infection, you go down, you're not able to work, right? So that's an indirect cost of antimicrobial resistance. So you take longer to recover, if you recover at all. And then there's the additional cost of extended therapies, right? Yeah, so like I said, there's both the direct mortality cost of resistance and the indirect or financial cost of resistance, right? So antimicrobial resistance, I like to show these little headers from newspaper clippings and online posts and whatnot about the seriousness, just to show that is not a by the way problem, is a relevant problem, and it's something that's growing every day, every year, right? And it's something that dominates news. Anyway, so we had a pretty good time discovering novel antibiotics early in the 20th century, right? So we had most of the antibiotics that we use now developed back then, discovered and developed back then. The problem is those were what we call low-hanging fruit. So You isolate a microbe from the soil, you find a bacteria or fungi that was active, you isolate the compound that it produced, and then you develop that, right? The thing is, over the past few years, there isn't much coming through the development pipeline, or the discovery pipeline, right? We have a couple new ones now, but those haven't been properly developed. And chances are they may not reach market because the classes of compounds they had to manipulate. The Tixobactin one is one that is relatively novel, but it's a very big molecule. And it's a proteinaceous molecule, so it's peptides. And the thing is, peptides are a little bit notorious to work with because in order to increase the efficacy or increase the safety, you need to modify these compounds. And sometimes a large molecule isn't the best, even though it has intrinsic activity, isn't the best to take through your developmental phases, right? Because they may have some off-binding effects and all of that. So now it's a very difficult thing, and people are now, researchers using all sort of weird avenues to, pretty innovative avenues to try and overcome the dried up pipeline of antibiotic discovery, right? And what I lead into is my research, and the thing is, or the question is, what if we didn't need to necessarily discover new antibiotics, right? With we could bypass the need to discover new ones, right? And it came about from an observation where early in my research, I realized that, and we knew this from before, that bacteria communicate with each other. And this is going back now to the beginning of the talk, where I said natural environments use chemicals to communicate. So in nature, organisms use chemicals to communicate. Humans develop speech, so we could communicate orally, right? But in nature, microorganisms, they can't speak to each other, right? Literally, but they figuratively do that through chemical interactions, right? So bacteria A will produce a compound that would elicit a response from bacteria B, right? Try to change the slide. Right. So what I noticed was that the bacteria I was studying was Pseudomonas, and it produced a compound that influenced the responses of other bacteria. So influenced the responses of some pathogenic bacteria, right? What it did was, it showed initially, it was just an observation, it showed that the particular compound that the bacteria was producing didn't allow the competing bacteria to form biofilms, which is a main step in infection in pathogenic bacteria, right? So the hypothesis was that, and we had named that compound SPBIM, right? Yeah, so the hypothesis back then was that the bacteria, my study bacteria, would have produced that compound to affect how competing bacteria would have responded to its own antibacterial compound, right? So then with development, or with isolating the compound, identifying it, and doing some further work, we realized that the compounds were known as antibiotic adjuvants, right? So yeah, so based on the activity that it displayed, right? I don't know if you all read it from the slide before. The compounds had no antibacterial activity, right? But what it did was, when we co-administered it with an antibiotic, it made that antibiotic that was ineffective, it made that antibiotic effective against a particular bacteria, or particular types of bacteria, right? And that type of molecules are termed antibiotic adjuvants, right? So they're sort of helpers of antibiotics. So the thing is that it breaks the bacteria's resistance to an antibiotic. And like I put there are 3 types of antibiotic adjuvants, so we don't need to get into that, right? So in developing the compounds, right, so I did some chemical modifications of the the SPBIM1, right? And I made about 40 or 50 different derivatives and tested all the derivatives, right, of the SPBIMs. And I got a couple of pretty good hits. So one of them was SPBIM13. And what SPBIM13 was able to do was lower the concentration of ampicillin, right, and other antibiotics, but the example here is ampicillin, right? It was able to lower ampicillin's potency, increase ampicillin's potency in killing MRSA cells. So MRSA is methicillin-resistant Staph aureus. It's a notorious infection, a notoriously hard to treat infection as well, over 100-fold. So the MRSA is resistant to ampicillin, but when combined with SPBIM13, we saw a 100-fold decrease in the MIC, or the lowest concentration needed to kill the MRSA cells, right? Dropping from 250 micrograms to about 2 micrograms, right? And this picture here is something called checkerboard assay, is what I do in vitro to test the adjuvant activity of the compounds. So I extended that activity to target gram-negative bacteria, right? So on the World Health Organization's list of bacteria by which antibiotics are needed, right? So they rank the infections based on priorities, right? So highest priority is Enterobacteriaceae, right? So that's Klebsiella, E. coli, stuff like that. Pseudomonas is another notorious infection. We have Staph aureus as well, Enterococcus. So all those different things that causes a whole host of different bacterial infections, and they're resistant to multiple antibiotics. So the picture on the screen here was a next hit called SPBIM126, and I used that to treat Pseudomonas infections. And the Pseudomonas was effectively resistant to a couple of different antibiotics. So ampicillin for sure, and ciprofloxacin in the diagram underneath, right? And what we observed was that SPBIM126 was able to lower the susceptibility of the Pseudomonas to below what we call a breakpoint. And breakpoint is what clinics use to determine if a bacteria is resistant or susceptible to an antibiotic. If it's below that concentration, if you get that below a particular concentration, it's susceptible. If it's above, then it's resistant. Essentially, what we did was restore the Pseudomonas' susceptibility to different antibiotics, right? And the activity spans a whole host of bacteria, and we get rejuvenation of multiple different antibiotics, which is remarkable because we have maybe 1 or 2 adjuvants on market, right? So you're maybe familiar with the beta-lactamase inhibitors like clavulanic acid, and I can't remember the others, right? Those only work against one particular mechanism of resistance, but these SPBIMs seem to have a broader range of broader range of adjuvant activity, right? I translated the in vitro work to some clinical isolates. So we got clinical isolates from infections across Trinidad, right? So these were from the hospitals. They gave us the bacteria, and we tested it. So these bacteria would have had different resistances against different antibiotics, and we did combinations with all of them. And in each case, the MIC, or the concentration needed to kill the bacteria, is circled in yellow, and the column next to it, encircled in red, is what the MIC was reduced to with a combination or with a co-treatment of the SPBIM, right? So I did that for clinical isolates, a whole host of clinical isolates, and in each case we saw a reduction. I then moved on to in vitro trials. Yeah, I then moved on to in vivo trials, sorry. So I took what we got in vitro and then translated it into mice studies. So I did two types of mice studies. I did a non-lethal infection where infected them with the bacteria and then monitored bacterial load in different organs. And then I did a lethal trial or a mortality trial to see if the compounds would be able to save the mice from death, right? And that was the general timeline of the treatments. So for the non-lethal trials, we saw in MRSA, we saw the SPBM13 was able to reduce we were able to reduce the bacterial load. So in blue, the bars in blue and green represents untreated mice, so mice that didn't receive any treatment, and in green, the antibiotic alone, which was ampicillin in this case. So in both cases, both the liver and the spleen, we saw high bacterial loads, right? But the bars at the base, right? Well, one of the bars at the base, the red bar at the base, was a combination treatment between ampicillin and SPBM13. So we saw almost total clearance. So there was no bacteria, almost no bacteria in the organs with the co-treatment, right? The gray bar next to it is a positive control. So that was vancomycin. The bacteria was susceptible to vancomycin. So we needed to compare that treatment, so to compare our co-treatment with a positive control. And in the yellow bar at the end, that was the compound on its own. And like I said, the compounds do not possess any inhibitory activity by itself. So we still saw a high load of bacteria, right? This one was against Klebsiella. Now, Klebsiella pneumonia is a pretty hard to treat infection, right? And it causes pneumonia in the lungs, as well as UTIs and whatnot. So I did the same sort of trial, and in this case, I looked at lungs as well. Apart from liver and spleen, I looked at the lungs, and I tested the activity in combination with two different antibiotics that the bacteria was resistant to. So it was resistant to ciprofloxacin as well as meropenem, or broadly class carbapenems, right? The trend was the same, that untreated and single antibiotic treatments had relatively high or very high bacterial loads in the organs, but the combination treatments resulted in almost a total clearance of the bacteria from the body, right? Right. Then in the lethal trials, which was interesting as well. So I infected the mice with a dose of bacteria high enough to kill them over time. And in this case, the graph on the left was SPBM13 versus MRSA, and then the graph on the right was SPBM136 versus Klebsiella pneumonia. And we see here that the mice that had the co-treatment, which is the lines in red, had 100% survival, right? We didn't get 100% survival, but we got 90 and 80% survival with the co-treatments against the Klebsiella, right? We had a couple of them essentially dying after one day, but most of them survived, which was significant because those that received single antibiotic treatments and no antibiotic treatment, they all died after 24 hours, right? So that was significant in itself, right? Our next phase of development was toxicity. So I studied toxicity and determined that all the compounds were tolerable above 2,000 milligrams per kilogram, which is the highest allowed level to test acute toxicity at, and they all survived, they all were fine, right? So the compounds then showed a good safety profile. Apart from mortality, when treated with high concentrations of the compound, we looked at their health, and across the span of 14 days, which was the observation period for the mice in the toxicity trials, they all seemed fine, they all were fine, they all were alert, calm, they had a normal appetite. So like I said, they had a normal appetite here, right? Monitored that daily, and then their weights over the 14 days were monitored every 2 days, and there was no significant differences between the treatments, which meant that was great, right? You don't want them losing a significant amount of weight, which would have translated into some sort of toxic effect. And then appetite too is almost direct correlation to some sort of distress, right? I did some druggable properties of the compounds, and it showed that all the compounds that we designed and tested were good candidates to become drugs. They fall into categories that make them good drugs, right? One of the main things is that they don't have any toxic metabolic products when processed in the liver. And these are unfortunately predicted.
Did we lose him?
Yeah, I think so. I'm gonna call him now.
Oh, okay, I'm just making sure it wasn't my internet that crashed.
Sorry?
Oh, okay. We lost you for a second there.
Why?
Like
Oh,
for
y'all did?
Yeah,
for
last thing we
half an
heard
hour.
was, yeah, like when you said predictable on the last slide.
And it was in the slide.
Yes,
Right.
yes, when you talk about the metabolic products, yes.
Right. So what I'm saying is, what I was saying is that unfortunately these were in silico predictions of the compounds to determine their druggability. So we want to move on. The next phase of the research is to move on into preclinical pharmacokinetic trials to determine if the predictions in druggability actually works. So we're doing some animal trials to see where the compounds distribute in the body, how it's processed, in the body, how long was the half-life within the body, where it accumulates. Like I said, so what organs it accumulates in, if it actually does accumulate at high enough plasma concentrations to warrant effective drug. So stuff like that is what we're moving on to next. So we have evidence that it works in vitro and in vivo. We have evidence that it's not toxic, right? We have also evidence that it will make good drugs, but we just need to prove that it does fulfill all the druggable characteristics. In determining how it works, we saw that it effectively downregulates genes involved in resistance and infectivity of the cells. So the ones circled in red are the ones I was interested in. This is called a gene ontology chart. So it gives you all the types of pathways that are affected or downregulated when the cells are treated with the SPBIM as opposed to the antibiotic alone. So we see that the bacteria is can't infect. It can't produce the resistant compounds or the resistant enzymes, etc. We also see they can't secrete those enzymes upon or toxins upon treatment as well, right? In response to the in response to the BIM treatments, the SPBIM treatments, right? This is just a gene regulation chart, right, to show upregulation or downregulation of certain genes involved in resistance, virulence, and global regulation of genes that determine how the bacteria responds to the environment. All right, and we saw with ampicillin, we saw upregulation of most of the genes, and with the compound, we saw downregulation of most of the virulence, deregulatory, and resistant genes, which means that the bacteria doesn't produce resistant doesn't produce resistant proteins. It doesn't produce the toxins and the pathogenicity factors that would make the bacteria virulent in the body, right? And then I confirmed that using something called real-time PCR, just to show that there was downregulation in those genes, right? In a whole host of resistant genes as well. And then it all led back to showing that what they actually target, or what the compounds do target, is something called a sensor histidine kinases. So this is what the bacteria uses to sense and respond to environmental signals, right? It spans the cell membrane, right? So a signal molecule would usually bind to the extracellular portion, and then a signal will pass down, causing ATP to bind to this catalytic binding domain, And essentially, it just allows the bacteria to respond. So if the stimuli is an antibiotic, it is sensed in the extracellular domain, and then genes involved in resistance would be turned on, and that allows the bacteria to produce the resistant proteins, the beta-lactamases, the carbapentamases, etc. What the compounds do is that, pay attention to the ATP binding site there. It binds to that ATP binding site, so it effectively outcompetes ATP for that site, and it doesn't allow the mechanism to occur downstream. So we get turning off, or we get essentially switching off of a lot of genes that the bacteria uses to respond to antibiotic treatments, essentially turning off the resistance and turning off the infection, the infectivity of the pathogen, right? So that's where I am at the research, right? Right now we're exploring avenues to to get funding to do the pharmacokinetic modeling or the pharmacokinetic trials, the studies. Essentially, we want to have a pretty good preclinical development package so that we could take to pharma companies and say, this is what we have, and they would take it forward into clinical trials to eventually, hopefully, become a drug on the market to treat bacterial infections. That's my contribution, guys. Thanks so much for listening.
Wow, thank you so much. This was such a great presentation. Very in-depth, and yet you were still able to bring it to a level that even if you don't have a huge understanding in pharmacogenetics, you can still understand it. So thank you very much for such a great presentation. I have a few questions, if you don't mind. First of all, congrats on getting this far into the research. This is really amazing, and the fact that you're able to show so much data is great. That's great. I love one of the gene ontology chart that you made is really cool. I've never seen something like that before. That was very interesting. First of all, how did you even make that? Like, is it something that you created yourself, or was there a program or something?
So it began with RNA-seq. So there's a bioinformatic pipeline, but essentially, when I got the upregulated and downregulated genes through transcriptome profiling and whatnot, right? And I used the Galaxy platform, Galaxy pipeline to do the to get the upregulated and downregulated genes, and I entered the up and downregulated genes onto a gene ontology. I think it's string database or something like that. They then identified what pathways were being affected broadly. So I did that gene ontology one, but then I could do KEGG. They have a KEGG database setting as well, so I could do KEGG. I think PFARM as well as option. So I'll do different ones, but this one was probably the easiest one to do. The pipeline is straightforward. It's from Galaxy, but once you get the Galaxy pipeline finished, then you just take the data you get based on significantly up and down regulated genes, and then enter it into StringDB. I think it's StringDB, yeah. In fact, I know it's StringDB, sorry.
That's really cool. Yeah, and I think the data was very well represented, so you can just easily visualize everything. Yeah. I had a question more so about the licensing of the IP, I guess, because you said you're looking at trying to seek funding from some pharma companies afterwards, right? So what's your plan with that? Do you have an idea of like how you would like to proceed in terms of like financially?
Well, yeah. So we have a patent for it already. So we filed that, and then so we're hoping we're going into national phase filing soon. So it's available on the WIPO website, the patent itself. But then eventually, yeah, so come soon, I think it's in May or June, you need to file in the individual countries under the PCT format. And that's going to be a little bit expensive. So our hope was to have a pharma company license it from us first so they could pay the, but I think we will pay. We'll obviously file in the major countries, the US, the Europe, US, Canada, Europe. So the main pharma markets, we'll file there. you might have to put out some significant money to file, but eventually the payoff is once we essentially have protection already because the patent is filed. But yeah, essentially we want to have a pharma company license it from us first. We're looking at we have a couple of people interested. Obviously they want the pharmacokinetic, the safety profiling done before, because that's probably, we did all this research to prove all of these things, and if it doesn't pass the preclinical safety, then it just probably doesn't make sense. Nobody would take it on, even though it is pretty well tolerated in mice and whatnot. But a mouse is quite different from a human.
That's great to hear that you have people who are interested already, and you've already got the WIPO patent, so you're moving forward with it. That's very interesting. Somebody's asked in the chat, Johnny Cyrus, what about the use of bacteriophages to kill these bacteria?
Yeah, so bacteriophage therapies are a pretty big thing, you know. But the problem is bacteriophage therapy in a clinical setting. So in an in vitro setting, so bacteriophages, for those of you all who don't know, is a virus that kills bacteria. So yeah, so in a lab setting, an in vitro setting, bacteriophages are quite effective. But when you administer a virus to in a clinical, so they tend to fail clinical trials because you don't get the same efficacy. So there has been companies that build platforms of bacteriophage therapies, but they tend to fail clinical trials because they don't have the same efficacy. Then essentially affecting somebody with a virus, and you hope the body doesn't kill that virus before it gets that bacteria. There's a bit of workaround, a bit of balancing, right? And so the couple of examples I know about bacteriophage development so far failed in clinical trials.
Right.
Yeah, that's some
question. So you were saying that usually the bacteria becomes resistant to antibiotics, right? Is there a chance that the bacteria may be able to become resistant to this treatment?
That's the question I always get. And the diplomatic answer is yes, you can't rule it out. So the domain, that ATP binding domain where the compound binds, is so conserved in bacteria that any change, so the only way it would become resistant to the BIMs is if it changes the protein sequence for that pocket, essentially changing the pocket that ATP would bind. But if it changes the pocket that ATP would bind, then ATP itself wouldn't be able to bind. So that conservation of that pocket makes it a good target for a compound to bind, because chances are it may or may not saying no, but based on the target of the compound, it's highly unlikely. But then you can't put anything past bacteria. So
okay, yeah, well, okay, yeah, that clears up like everything. But I understand what you're saying, that
you understand the point?
yeah, it would be hard for them to change their ATP binding site, because then they would have to either not use ATP or the ATP itself would have to change.
Yeah, exactly.
It's a good target spot. Okay, yeah. Well, that's some great research. I feel like if I were all smart after hearing this, you know. Yeah. Does anyone else have any questions?
Yes, good evening. I have a question about I think one of the experiments you did was with ampicillin. I'm not sure what the pathogen was.
Oh, MRSA.
But I think
yeah,
when you combine it with the adjuvant, you got like a 90% survival or something. No, the ones that were in 100% with the adjuvant, why would you say they didn't survive?
That's a good, yeah. So the ones that didn't survive 100% was Klebsiella pneumonia, and it was a combination treatment with, so there was two antibiotics I tested there. So one trial was with ciprofloxacin and SPBIM, and the other one was meropenem and SPBIM, and then meropenem and cipro on its own, right? The mice that died, essentially, we put it down to shock. they got such a high dose of bacteria initially that maybe themselves, the individuals themselves, wasn't able to recover, even though we gave them the antibiotic treatment. And the deaths that occurred all occurred after the first day. So we put that down to back to shock, but in terms of significant differences or statistics, it still gave us statistically significant higher survival rate than the individual antibiotic treatments, because they all 100% died after 24 hours.
Yeah, for sure. I mean, it was definitely a huge difference in effect. When you say shock, so weren't all the, like, there's a way, I'm sure you'll measure the amount that the rats are given in terms of bacteria, right?
Yeah.
So is it something, what would lead them to get the shock?
So yeah, that could be due to the individual mouse immunity then. So some mice might be able to withstand an initial infection better than some. So it probably was down to the individual mouse's response. If you understand?
Are they genetically identical?
No, they're not. They're not genetically identical at all. They're bred in lab, but to say they're clones of each other, no.
Right. My second question, third or whatever, would be would you say there's a risk to relinquishing some more level of control when you license a drug company than you would like, in terms of maybe perhaps things like the pricing of the drug?
Boy, that's a good question. Unfortunately, I can't answer.
That's big unknown.
Just repeat the question. Sorry,
okay. So like in my mind, I'm picturing two routes. You either, when you get the data for the preclinical data in terms of the pharmacokinetics, right? One of the routes I would say is if you get venture capital funding and you started your own company and then made a deal with the pharma companies, or you go straight to try to license to the pharma companies to do the trials themselves, for the companies to do the trials. So I'm just wondering, like, do you see a significant problem with you licensing it early? Do you feel like I'm just wondering what's that effect on you, I guess.
Oh yeah, so with a venture capitalist investment, essentially they give a certain amount of financing, but they expect a pretty big return.
Yeah,
right?
as you think too.
Yeah, so they expect a pretty big return. With going straight to licensing to the pharma companies, well, obviously we would have some sort of, and I guess that's a more legal question, a little bit out of my domain. Hopefully, eventually, one day, that's a good idea. Nita, y'all could probably have a IP person or a commercialization person come in and do some protection, or how you make money off some scientific thing. But yeah, but I know that aspect, that particular financial and legal thing is being taken care of by UE for us. So we don't really get involved in that because the invention is at the end of the day owned by UE. So they would decide the best route of advancement. And I think right now we at the point where UE would put out, or UE, I hoping that UE would give us the money to develop, and then we move to license, so out licensing. We don't really want to get, well, I don't think it's in UE's best interest to get funding from outside before to do these preclinicals, because then they're expensive, yes, but they're not an exorbitant amount of cash that the university can't probably put out. But the best route would be for UE themselves to pay for the preclinicals, and then we license that company to do the clinical trials. So we give the license of thing, obviously with profit sharing and IP sharing and whatnot, royalties, etc. Yeah, but from the time you're bringing a third party like a venture capitalist, then you essentially share the IP three ways, and then that sharing of the IP usually is veered towards the venture capitalist fund, because they set this is X we want in return for our seed investment. In terms of pricing is basically down to the drug companies who eventually get the license at the end.
The next question, you mentioned how UE they own basically the product. Is that clear? What you said?
Yeah, any research done in a university environment is, so you have the inventors, which would be me, but the IP itself, the product is owned by the university, because essentially they say it's their labs, it's their materials, it's their salaries, etc. So they own. So if you look at the patent on WIPO, you would see the inventors, but then you would see the patent is owned by University of West Indies.
Wait, so like if they have to make a decision, and when it comes monetary wise, you have like no percentage of that?
No, yeah, definitely. So before we go into any, if you have an so UE is pretty good like that. If you have an idea, a money-making idea or a money-making invention, you sign a non-disclosure agreement with UE first, and essentially you, the inventor. UE has a set thing in that they take 50% of whatever you make, and the other 50% is split between the inventors.
Oh, okay. Thank you.
It varies. I know it varies between universities, but for UE, that's their set in stone template.
Okay, thank you very much.
Yeah.
Okay, so does anybody else have any questions for Antonio? Anything to ask him?
Am I allowed to ask him a question, or will he yell at
Will you yell at Antonio?
in his face?
Like, oh God.
I, as a member of
Hi, Antonio, I wasn't sure away.
How you doing? Long time.
you see what I'm talking about?
I believe the girl in orange had a question.
Yes, the girl in orange did have a question. I just had one question. Okay. The mice that died due to shock. How is that going to translate for actual human trials when the time comes?
So the level. So I want to repeat that study. So I'm going to repeat that study probably with a lower.
With the mice themselves, right?
Yeah, with the mice themselves. But you will never get a human being infected with 900 million bacterial cells per mil. So that's not the level.
Anything is possible.
So the level of bacteria that I inserted into the mice was way beyond what you would typically expect to get from a bacterial infection, unless it's pretty bad. So we're looking at terminal bacterial disease modeling there.
Why did that? Just like as a standard thing, you had to do that much, or you just chose to see how far you could
So what I did was, before we began the trials, would have given them different doses and see which dose would have resulted in death within 24 hours. I took the highest end there, so that's probably a little bit overkill for me, because I just wanted to probably make a bigger point or prove a bigger point. But I might need to decrease, either increase the concentration of the compound, the co-administered compounds, which I don't want to do. But I want to probably decrease maybe tenfold the amount of cells that are infecting the bacteria with.
Okay, that's it for me.
Okay, so anybody else, or?
Yeah, I have a question, Antonio.
Okay.
So at what point in time did you realize that this is going to be a lifelong career pursuing antimicrobial compounds from Pseudomonas, right? Because I missed half your presentation.
Yeah. So yeah, it began, but I don't want to stick to the Pseudomonas. I want to move on. So you missed the first half. I was talking about metagenomics. So that's the direction I want to head into. Because this discovery was good, but to be honest, is pretty much down to rare luck with some hard work. But yes, but I want to move forward into probably different. So I want to get this done, get this developed, and probably onto clinical trials, and then move on onto to more discoveries and different things, but stay in the realm of antibiotic discovery.
Cool. And 10 years ago, when you now started this, you would have pictured yourself down this line and being where he is right now?
No, I didn't. I mean, no, I knew I'd like research, but I didn't think I would have reached where I am now. No, I didn't think, because to be honest, you have your plans, but then when something takes over, you just continue with it.
Because I've seen a lot of young budding scientists up and ready. How are we going to keep them on the path? I didn't follow the path. I do apologize.
I know,
let
if you
you
know,
down.
this was the first person that I was microbial biotech.
Yeah.
Yeah, it was the first guy from the group I had. He didn't know how to swim, and somehow now he's a diver.
Yes.
Yeah,
But
I think
just along those same lines, we have some like undergrads in here and like maybe some secondary school students. So like for those who are interested in kind of going along the same path, like how would you suggest that they get into research, especially being in Trinidad where there's not a lot of investment into it? Like what advice would you have to kind of guide them into going down that path, if you have any?
That's a good the thing is, the research isn't glamorous. So I presented there, and y'all probably watch that and be like, whoa, that's excellent research and all of that. But it takes a lot of small steps every day, and a lot of the times I would feel like I'm not moving along, right? So it's a pretty persistent. You gotta have persistence. You gotta know how to take hits and get back up and find a way to bypass any hindrances. But then you gotta have a passion for it as well. And I think that's probably the biggest thing. Once you're passionate about it, you'll find yourself in the lab every day. You wouldn't think it's a burden to get up in the morning and come. I just get up in the morning, and that's the only thing I'm thinking about, coming and trying to do something or getting stuff done in the lab. And it pays off in the end, because I'll tell anybody, you don't need to be smart to be a scientist. You just need to be passionate, and you need to be able to think outside the box. Because a protocol, anybody could read how to do PCR or how to do DNA extraction. That's basic things where you're getting a paper in front of you and add ingredient A, shake, add ingredient B, do that. So everything now is pretty much straightforward in terms of the basic stuff. The thing is for you to be able to think outside the box and make sense or come up with your avenues to overcome a particular problem. And I think once you're passionate about something, like I like discovering molecules, right? And I wasn't a chemist. I didn't do chemistry in undergrad, but basically taught myself how to do NMRs and mass spec and everything else, because it was something that I was passionate about now. So like I said, yeah, you don't need to be the smartest person, because I definitely am not the smartest person. I am not, but you need to be passionate. You need to have that drive to get it done and to find a solution to your problem.
I see.
I think too now, it is a difficult avenue, but I'm due to people like Nita and Tevin and them guys. I think we put in science more mainstream in Trinidad than it was when I began. Nobody knew what scientists in Trinidad are. You have lecturers in UE, and that's probably as much as you would know, or as much as the public would know, but nobody would really know that there are scientists or research scientists around. But I guess with more publicity, people would realize that research is occurring in Trinidad. Trinidad is a good place to do research, and now just to get the people involved, or people in policy making and people with the money, the government, to be a little more enthusiastic about science and to probably dedicate more resources into science and tech, because that's the only way our industry can move forward. Our nation can move forward.
Yeah, I agree, because there's like so little, like other countries are now moving towards like precision medicine, and we still kind of stuck on trying to perfect modern medicine, and it clearly not working. So, and there's so much potential locally as well. Like you said, with the bioprospecting, there's so much diversity locally that I don't think we're taking full advantage of it.
Yeah, I think people come to Trinidad, and take our resources and then go and find great things.
Yeah, a lot of people come to Trinidad, especially for like guppy research and a lot of organisms. We have a vast biodiversity, so we should probably put it to use. So thank you, Antonio, for starting to put it to use.
Yes.
I can say for sure that people do, because just as Stefan was talking about with that guppy research, right before lockdown last year, actually, one of my colleagues from University of Bristol, she went down to Trinidad to do guppy research. So I know for sure that people do come to Trinidad to do research, and it's kind of infuriating, that people are coming and kind of taking our raw material, so to speak, to go and do research when we could be doing it ourselves. But I do want to say thank you so much, Antonio, because it's very refreshing to hear somebody from Trinidad speaking my language, because as somebody who's studied abroad like my whole life, I haven't really come into contact with any other Trini or the Caribbean scientists in my field. And so that's why I'm very happy that we got to organize this journal club, because I've been waiting to hear somebody speak my language, so I'm very glad to be in a community like this, and hopefully we'll grow and we'll get some more people in as well, because it's so refreshing. So thanks a lot.
My question is, I'm not too familiar with how the immune response is for bacteria, but if we could find like good targets to create like monoclonal antibodies, do you see that as a promising pathway as well, to try to
antibodies to target bacterial infections? Well, I think there are, what is this vaccine for? You're accustomed getting tetanus shot, right?
But wouldn't you say, like, I mean, because you could have a vaccine that doesn't produce a high immune response, right? But is there a way then that instead of giving them the vaccine, you just give them the antibodies, like a high amount that the common vaccines aren't producing? If that makes sense.
That's a different level question there, boy. So the thing is, the antibody itself, you see, the antibody would need to be accepted by the body. And because it's a protein, you don't know if the body itself might just break that down, or if it does get might next thing elicit a response from the body, because it's a foreign antibody you're putting into your body now. I'm not saying it is not possible, but antibody treatment for bacteria, it could work, because you could develop antibodies for some of these surface proteins where the bacteria would bind, that would use to bind to cells to infect the cells. And I think that's an actual thing that people are looking into, to develop antibody therapies to bind to membrane proteins on the bacterial surfaces, that would prevent the bacteria from infecting cells. Because in order for the bacteria to become infected, most of the time it has to go into a cell, and then it will produce toxins to burst the cell open and all of that. So yeah, but that's a good question. That's a pretty good thing. I think I'm not sure, but I think people are looking into vaccines for those things. Well, antibodies for those things.
What I saw was just like how it's kind of easier with COVID because of these surface proteins. Like for certain things, I guess, I don't know if it's because like they change or whatever, that like if you target like the surface, it might not work. Like you have to find a way to target something that's universally, would be universally read, so to speak. I think that's what I saw was one of the problems in terms of targeting the right surface protein to attach to bacteria or whatever.
That's true. I mean, I guess bacteria, it it will change with the species. It'll change. The surface proteins will also change with subspecies and mutations and whatnot too. So yeah, that's a big problem to overcome. That's a pretty big thing to overcome, and pretty variable as well, like you were saying. I'm not sure if you will be able to develop a antibody that would target universally all attachment proteins for all bacteria, but I mean,
that would be wild.
Yeah.
I think that's kind of the issue, right? Because I know there are definitely like antibody treatments for like cancer, for example, right? But it's much easier when you're targeting a specific cell. So for example, I'm trying to remember, they target a specific protein on the surface of breast cancer cells. I think it's the HER2 gene. I could be wrong. But basically, the idea is that it has to be something that's quite specific because it is an antibody at the end of the day. So that it's very specific binding. So to make a cure-all might be quite hard. Also to make it to attach to something external as like an antigen, I think that makes it even more difficult. So it might be possible, but maybe not necessarily for antibacterial purposes, I think.
Yeah, that's true. I don't think we need to. I mean, we may eventually need to get that extreme, but then chances are if you're looking to target something inside the cell, how is the antibody, how is that protein going to get in the bacterial cell itself? And like you'll just pass through the membrane. Bacterial wall is in place there. You have pumps. You have all those selective things that bacteria are just not allowed allow that protein especially to get into the cell. A small molecule, like an antibiotic, those compounds are pretty small, so you could sneak through pretty easily, but something as large as a protein and even bigger as a antibody, then that's probably not ever going to get into a cell, a bacterial cell.
Yeah, I think it makes more sense as a prophylactic vaccine-wise. That makes more sense. A better target for vaccines. That would make sense.
Okay, so has everybody gotten in their questions? To Antonio, to everybody, last call. Okay, I guess that is it. Yeah, I'm not seeing anybody else asking anything. Seven, do you have a question? Your mic is just on. Okay. All right, somebody said it was very informative. Antonio, Kriya said to let her uncle, that's you, know that you made her very proud. And somebody said your research is really cool as well. So everybody loves your research.
Right.
Okay, so thanks for coming. Thanks for being our first presenter and everything. And thanks for letting us record it. Oh, there's somebody else message in the chat.
They said great presentation.
Yeah. So who you're looking at next? Any who's going to be the second?
Well, I see Jude's here. I'll ask you, Judy.
Yeah. Also, we have the guy from New Zealand who's doing the seismic. Was it no? Communication.
Is that for this or is that Tevin? I'm getting confused.
Yeah, it might be. It's okay.
Basically, short answer, it's in the works.
Yeah. We have a few people lined up. It's just to get what?
How often you're looking to do it?
Every other week.
Oh, so relatively often.
Yeah. We would try.
Yeah. I think you'll have a lot of people who would be interested.
Yeah. So we want to like hit up all the different disciplines and whatever. And then we have a few people internationally and regionally, thanks to Darian. So yeah.
Right. I agree.
All right. So I think we could wrap up now because Darian is in the UK. So he's way past my bedtime. So yeah, thanks for coming on, everybody. Y'all can look out for the emails for the next session as well. As long as you use the Google Forms to sign up for it, you get the emails whenever we have someone lined up to present. Okay, so yeah, thanks Antonio and thanks everybody else for coming. Have a good night. Bye.
Bye everyone. Thank you.
Bye everyone.